Hi, my name is Hugh Gehring, and I'm presenting for the University of New England Medical Biochemistry 1 class for the Unit 2 Case Connections. I've chosen to present on Case 1 on the topic of pyruvate kinase and glycolysis in cancer cells, and Case 2 on the topic of glucose uptake in a glioblastoma. Case 1 is as follows. Pyruvate kinase catalyzes the rate-limiting ATP-generating step of glycolysis, in which phosphoenolpyruvate is converted to pyruvate. Multiple isoenzymes of pyruvate kinase exist in mammals. Type L, which is found in the liver and kidneys. Type R, which is expressed in erythrocytes. Type M1, which is found in tissues such as muscle and brain. And type M2, which is present in self-renewing cells such as embryonic and adult stem cells. Under normal conditions, describe all the ways in which pyruvate kinase is regulated in the cell. Well, pyruvate kinase is regulated by allosteric inhibition via ATP and alanine. Alanine is transaminated from pyruvate, and both indicate excess product. Gluconeogenesis and the favoring of gluconeogenesis rather than further glycolysis. It also is inhibited via covalent modification via phosphorylation catalyzed by cyclic AMP-dependent kinase, which is activated by increased cyclic AMP levels, whose production is stimulated by glucagon and epinephrine. Pyruvate kinase is also limited by substrate availability via upstream reactions, such as issues with insulin-triggering glucose transporters to initiate glucose uptake, like in diabetes, hexokinase, another regulating step in glycolysis, and phosphofructokinase 1, which further phosphorylates fructose 6-phosphate to fructose 1,6-bisphosphate. Pyruvate kinase is allosterically activated by fructose 1,6-bisphosphate, which is achieved via feed-forward activation and increases the flux through glycolytic pathway. Pyruvate kinase is also activated by covalent modification, the dephosphorylation, undoing the effect of the cyclic AMP-dependent kinase. Continuing with Case 1, many types of cancer cells are predominantly glycolytic and overexpress the oncoprotein MYC. Interestingly, the classical oncoprotein MYC has been found to promote preferential expression of pyruvate kinase type M2 over pyruvate kinase type M1. The isoform M2 is characteristically found in low activity state and is ineffective at promoting glycolysis when compared to pyruvate kinase type L or type M1. This logic seems counterintuitive for glycolytic tissue. What is the rationale for this mechanism, and what other pathways may cancer cells be diverting substrate to? So let's break that up into two steps. What is the rationale? The isoform M2 of pyruvate kinase is present in self-renewing cells. It may be that the higher activity and proliferation that is stereotypical of cancer cells would be enough to term them a type of self-renewing cells. It would then make sense that cancer cells overexpress a protein, the oncoprotein MYC, that promotes expression of the pyruvate kinase in self-renewing cells, as that is what they are. Cancer cells primarily use glycolysis for energy rather than oxidative phosphorylation. Being ineffective at promoting glycolysis, it may also be that PKM2 is needed in excess to overcome the low-energy glycolytic pathway and to compensate for the high energy demands of rapid-growing cancer cells. The combination of high energy need and high number of low-effective enzymes expressed explains the enormous glucose consumption that is typical with cancer cells and malignant tumors. Cancer cells have rapid growth and proliferation and need higher number of glycolytic intermediates to be used in various other pathways for their prolific rate of cell growth. The ineffective isoform M2 leads to excess buildup of intermediates to serve this function. This is why the ineffective version of pyruvate kinase is needed. This effect is called the Warburg effect. Warburg effect describes how cancer cells favor metabolism via anaerobic glycolysis pathway despite aerobic conditions. permitting the more efficient and productive oxidative despite aerobic conditions permitting the more efficient and productive oxidative phosphorylation pathway. If they produced a different isoform, this buildup would not happen, and the effect would be lost, leading to lower rate of proliferation. The Warburg effect leads to increased substrates for the synthesis of other products, and also the resources that allow cancer cells inhibition of macrophage actions and to avoid apoptosis. Other pathways that the cancer cells may be diverting substrate into. Well, the intermediates become substrates for synthesis of nucleotides, ribose synthesis, like in the pentose phosphate pathway, protein synthesis, and cell membrane components. Let's move on to case 2. They're very similar. Regulation of glucose uptake by the cell. The above MRI shows a large glioblastoma tumor with areas of necrosis within the tumor. The tumor is the bright white area within the brain field on the MRI. On the right is a PET scan from the same individual following administration of fluorescently labeled D-glucose. Use the following series of questions to determine how or if glucose uptake differs in the tumor when compared to the surrounding tissues. So, in normal tissue, how is glucose uptake regulated? It gives us 4 types of tissue: liver, skeletal muscle, brain tissue, and adipose tissue. Liver is insulin-independent glucose uptake. The cells are freely permeable to glucose via glucose transporter, and glucose is trapped in the cell via phosphorylation by hexokinase and glucokinase. Glucokinase is the non-insulin dependent isoenzyme of hexokinase with a lower affinity for glucose. The GLUT2 transporters are always present on the cell membrane. This is what allows the cells to be freely permeable to glucose. Skeletal muscle and adipose tissue are very similar in that they are insulin-mediated glucose uptake. Hexokinase again traps glucose in the cell after transport into the cell by insulin-dependent GLUT4. Insulin triggers a protein cascade response which activates the GLUT4 transporter to come and bind with the cell membrane, allowing for glucose to be transported into the cell. In the brain, we have hexokinase again trapping glucose in the cell after transported into the cell by non-insulin dependent GLUT1 and GLUT3 transporters. The transporters are always present on cell membrane. GLUT1 mediates the transport of glucose across the endothelial membrane, the blood-brain barrier, and uptake from the extracellular fluid. GLUT3 facilitates neuronal glucose uptake. So how does glucose uptake in normal tissue compare to glucose uptake by tumor cells in the above image, and why would this be advantageous in the invasive tumor? So in the PET scan, the tumor is very fluorescent due to the fluorescently labeled glucose and indicates a much higher rate of glucose uptake from the surrounding tissue. The Warburg effect that we spoke about in case 1, where cancer cells favor anaerobic glycolysis pathway despite aerobic conditions permitting the more efficient and productive oxidative phosphorylation, does not need apply only to situations where aerobic conditions are permitted. It allows for survival in hypoxic conditions, such as how the large glioblastoma is growing against the skull and likely occluding surrounding capillaries or arteries to that area. This allows, the Warburg effect again allows for increase in intermediates and for use in cell growth and proliferation, and the glucose-heavy uptake and glycolytic pathway usage by cancer cells allows for continuous energy production and the production of these intermediates. Despite being swollen mass of cells occluding capillaries that provide glucose, tumor cells get glucose via GLUT1 facilitated transport across the endothelial membranes. The large blood-to-brain concentration gradient drives the facilitative transport of glucose across the endothelial membranes via GLUT1 glucose transporters into the extracellular fluid, whereas GLUT3, which has a much higher transport rate than GLUT1, facilitates the neuronal uptake of glucose. Based on the continuous growth of the tumor and the fluorescent labeling of the tumor in the PET scan, I think we can indicate that the tumor cells, the cancer cells, have a higher rate of glucose uptake than the normal tissue. What modifications may have occurred in cancer cells to alter glucose uptake as predicted above? Glioblastomas typically have a GLUT1 overexpression, leading to even more glucose uptake into their tissue. Brain matter relies heavily on glucose for its metabolism, and this makes it particularly difficult in treating because you cannot limit the glucose uptake of the surrounding tissue, as the brain matter relies heavily on it as well. There's also M2 pyruvate kinase isoform overexpression, as we spoke about in the case 1 connection, that allows for the utilization of glucose in a way that provides many intermediates for various other biosynthetic processes. Thank you for watching.